In this video, we will discuss about out of this IL27R alpha and this IFN gamma plus, which cytokines is responsible for anti-tumor response in the STUB1 and CHIC2 knockout in CD8+T cells. Though from the heading it is clear that it's the IL27R alpha is responsible. So how they come up to this conclusion, that we will discuss in this video. In order to check, so their hypothesis is to check whether both of these cytokines are responsible in the anti-tumor response or only one of them is responsible. So what it is, they took this naive CD8+T cells and transduced them, or nucleofection is carried out with knocking down this STUB1, IFN gamma R1 and this CHIC2 knockdown and this IL27RA knockdown, and transduce them in this B16 OVA tumor injected mice. Or once this nucleofication is done, then this is transduced into the mice that has this B16 OVA tumor. And so what they are able to find out is that compared to the control in the STUB1 knockout that we seen in the previous video, that the CD8+T cells goes up, but when this IFN gamma 1 R knockdown is used, do not see any fold change compared to the control. But when there is STUB1 knockdown plus IFN gamma 1 R1 knockdown is there, then they are able to see significant fold changes, indicating that IFN gamma 1 R does not play an important role. Even if it is knocked down in the STUB1 knockout, the fold change does not change, neither increase nor decrease, as p-value is 0.15. And when I check it, did the similar studies with IRA alpha knockdown. So for the control, they see that the fold change is low, but when the STUB1 knockdown is there, and along with that IL27R alpha knockdown is also there, they do not see too much increase in the fold change, indicating that this IL27R alpha plays a role towards this anti-tumor response or its proliferation of CD8+T cells. And when they so once this is like in indirect validations. For direct validations, they check the cytokines that is responsible for apoptosis are released by T cells or not. For example, granzyme positive, perforin positive, and they also check for IFN gamma positive, and they can see that all these are increased in the STUB1 plus IFN gamma R1 knockdown more significantly compared to the case where STUB1 plus IL27R alpha knockdown is there. And for IFN gamma plus also they can see the IFN gamma plus is even more than the control in the IFN gamma R1 knockdown and STUB1 knockdown compared to the IL27RA alpha knockdown. And the full norm, and when they did the similar studies with CHIC2, they find out the similar results, that when the IRA alpha knockdown is there, they are able to see a minimum fold change, and when this CHIC2 plus IRA2 alpha knockdown is there, they are still able to see minimum changes, indicating that this IL27R alpha plays a role towards anti-tumor response or in enrichment of the CD8+T cells. So once validating their results finding out the results in the in vitro studies, they wanted to validate their in vitro results in the in vivo studies. So they first check it in the mice with STUB1 knockdown, and then also this IL27R alpha knockdown is also there, and in one case both the knockdown is there. So in the control, they can see the tumor volume increases, but when this STUB1 plus IL27R alpha knockdown is there, they can see tumor volume increases more significantly, and when only this STUB1 knockdown is when only this IL27R alpha knockdown is there, the increase in the tumor volume is more steeper compared to the case when only this STUB1 knockdown is there, where tumor volume does not increase much, indicating that this IL27R alpha plays an important role, and it also helps in reductions in the tumor volume. And when it comes to the percentage survival, there also they see the similar results, that with STUB1 knockdown they are able to see maximum survival rate, whereas when the IL27R alpha is also knocked down, survival rate goes down. And when they did the similar tumor volume and percentage surviving studies with CHIC2 knockdown, they can see the similar results. That is, when CHIC2 knockdown is there, they are able to see a delayed tumor growth rate, and in the control they are able to see a steeper growth rate. But when this IL27R alpha knockdown is there, the growth rate further increases, and when this CHIC2 and IL27R alpha knockdown is there, the growth rate is more steeper. And similarly, since the tumor volume goes up in a short duration of time, the survival rate also goes down in the case where in the case where IL27R alpha knockdown is there, or CHIC2 plus IL27R alpha knockdown is there. And for the control also, since in that case STUB1 is present, the survival rate are low compared to the case where no STUB1 the CHIC2 is present, survival rate is low compared to the case where CHIC2 knockdown is there, the survival rate is maximum. So all these results in together pointing out to the fact that IL27R alpha plays an important role in the anti-tumor response in either STUB1 knockdown or and in CHIC2 knockdown in CD8+T cells. So that's all for this video. I hope you understood this story about the STUB1 knockdown or CHIC2 knockdown with this IL27R alpha in the CD8+T cells generate anti-tumor response. So this is the last main figure of this paper. If you need to understand more about this story or these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.